Interleukin-18 is a key cytokine that, when combined synergistically with interleukin-12, has the potential to be an important therapeutic asset for oncology and cell-based therapy.
Welcome back to another Virtual Investor What This Means segment. My name is Janine Thomas, and I am CEO of JTCIR. Today, we are very excited to feature Sonnet BioTherapeutics to Virtual Investor, and I'm very pleased to be joined by Dr. Pankaj Mohan. He's Chief Executive Officer, and Dr. John Cini. He's Chief Scientific Officer of Sonnet BioTherapeutics. Before we get started, I just want to inform our audience that Sonnet is listed on NASDAQ and trades under the ticker SONM. And during today's discussion, the company will be making forward-looking statements, and I encourage everyone to view the company's website at sonnetbio.com or the SEC's website for their latest filings and information. So, gentlemen, we're going to dive right in. Congratulations on this exciting news. So, for our audience, Sonnet has been issued a U.S. patent covering a variant interleukin-18 incorporated into two novel immunotherapeutic drug candidates. Pankaj, what does this mean for Sonnet?
Thank you so much, Janine. I am really excited. The issuance of this intellectual property for the interleukin-18 variant is an important milestone that we believe provides significant differentiation from any other competitive positions that may leverage the full biological potential of interleukin-18, either alone or in combination with interleukin-12. Interleukin-18 is a key cytokine that, when combined synergistically with interleukin-12, has the potential to be an important therapeutic asset for oncology and cell-based therapy. Further, interleukin-18 can be combined with synergistic biologics on our FHAV platform to create a number of entirely novel partnering opportunities. Finally, we believe such an important intellectual property from the scientific and clinical relevance standpoint adds significant intrinsic value to Sonnet.
Pankaj, could you provide us with a brief overview of interleukin-18 or IL-18?
Interleukin-18 has taken center stage in eliciting pro-inflammatory cytokine responses along with interleukin-12 or IL-12. We believe that receipt of this patent covering a novel interleukin-18 variant combined with our platform provides further differentiation, extends the half-life and biological activity of linked molecules by binding native albumin in the serum, and targets the tumor microenvironment. Interleukin-18 has significant importance for cancer immune oncology, and combination with interleukin-12 on Sonnet's FHAV platform, which I believe could present an important oncology possibility.
John, over to you. So what are the key aspects of IL-18 that make it a very important cytokine?
IL-18 can regulate both the innate and adaptive immune response through its effect on natural occurring NK cells, monocytes, dendritic cells, T cells, and B cells. IL-18 acts synergistically with other pro-inflammatory cytokines to promote gamma interferon production by the NK cells and T cells. Systemic administration of IL-18 has been shown to have anti-tumor effect in several animal models. Moreover, the tumor-infiltrating lymphocytes express more IL-18 receptor than other T cells do. However, in IL-18 clinical trials, it has been shown, though well tolerated, that IL-18 has poor efficacy in treating cancers. Most likely, this is due in large part to the high co-expression of IL-18 binding protein, referred to as IL-18 BP, in the tumor microenvironment. In particular, IL-18 BP serves as a decoy receptor that binds to IL-18 with very high affinity compared to the IL-18 receptor complex, thereby causing a negative feedback loop with IL-18 and inhibiting its mediated tumor infiltration T cell activation. Thus, there exists a potential for the discovery of an IL-18 variant composition that does harness the therapeutic potential of IL-18 for the treatment of cancer. Sonnet has developed a novel IL-18 variant that does not bind to the IL-18 BP inhibitor while maintaining biological activity. We believe this could harness the therapeutic potential for the use of Sonnet's IL-18 Fab construct platform for the treatment of cancer patients. As in many human cancers, it has been shown to have high levels of this IL-18 binding inhibitor. For example, head and neck, melanoma, kidney, glioblastoma, pancreatic, and stomach cancer.
So, John, I'd also like to ask, why do you think that the combination with IL-12 on your FHAB platform is a key differentiator?
The lock and load flexibility of the Fab platform offers the capability of creating bifunctional payloads with extended serum half-life and tumor targeting capabilities. Adding another synergistic biological target to IL-18 allows a generation of potential novel cancer drugs. Sonnet 1411, which is IL-12 and IL-18 in combination, is a proprietary bifunctional fusion protein consisting of the IL-18 binding protein resistant with the combination of a single chain IL-12 linking to the Sonnet Fab platform, which replaces the 1410 construct, which was under development target. Sonnet 1400 is a monofusion composed of the Sonnet IL-18 binding protein resistant domain linked to the Fab platform. IL-18 and IL-12 can act synergistically in the following ways. IL-18 in combination with IL-12 synergistically induces gamma interferon activation in NK, CD8, CD4 T cells, which upregulates PD-L1 on tumor cells. IL-12 upregulates the IL-18 receptor. IL-18 upregulates the IL-12 receptor. IL-18 increases chemokines called CXCL9 and 10 expression, which is increased in Th1s, NK, CD8s, and T cells infiltration into the tumors. In the presence of both IL-12 and IL-18, these chemokines 9 and 10 expression was increased 50-fold. IL-12 activates T cells and NK cells by reducing the immunosuppression conversion of M2 to M1. The Fab technology targets tumors, lymphatic tissues, providing a mechanism for dose sparing and the opportunity to improve safety and efficacy profile of IL-18 and IL-12, as well as a variety of potential immunomodulators that can add to the platform. IL-12 can orchestrate a robust and immunological response to many cancers and pathogens.
So, Pankaj, before we close, beyond IL-18, the company also has monofunctional and bifunctional candidates targeting other important cytokines, including IL-12 and IL-15. Can you give us a rundown of the rest of Sonnet's pipeline?
SON 1411 will be our second bifunctional asset. As you know, SON 1210, which is a combination of interleukin 12 and interleukin 15, is currently being developed by an academic consortium for pancreatic cancer. As you know, SON 1010, our monofunctional interleukin 12 asset, is currently being developed in collaboration with Roche as a combination with Atezo for the treatment of platinum-resistant ovarian cancer.
With that, this concludes the Virtual Investor What This Means segment with Sonnet BioTherapeutics. I'd like to thank Drs. Pankaj Mohan and John Cini of Sonnet BioTherapeutics for joining us today. As a reminder, Sonnet trades on NASDAQ under the ticker SONN. And if you like what you saw today, I encourage you to visit sonnetbio.com for more information on the company and to sign up to follow the company to receive their alerts, as well as follow their social channels to stay current on the latest information. You can also visit virtualinvestorco.com for our latest segments and events calendar. I'd like to thank everyone and wish everyone a great rest of your day.